Well, good morning, AP Bio students and teachers from Advanced Kentucky. This is Lou Akampora, and we are getting ready for our final content review session online in preparation for the AP Biology exam. Our presenter here today is Brittany Cox, and many of you know Brittany from Jessamine County High School. I'm trying to do a lot of things here, and she'll be talking about Mendelian genetics and heredity. I'm hosting you and with my colleagues back in the booth here. And then just a reminder for meeting etiquette, please do mute your microphones and turn off your video, but make sure you use the chat box, and especially this is a reminder to you students from Barberville High School. Ms. Hubbs cannot be with us today, but if you are to get extra credit, you need to log into the chat box. So please put your name and your high school in that chat box. And I might if someone will remind me to remind other students who might show up late to do that. Another reminder is that this session will be archived at kyap2020.com, along with any support materials from all the other sessions. So once again, Brittany Cox will be talking about Mendelian heredity, Mendelian genetics, and heredity. And I will turn it over to Brittany.
Good morning. I want to congratulate you guys for getting out of bed this morning in time for some science. I know that's not necessarily the easiest thing to do. All right. Okay. Does this look okay to everyone?
Looks good.
Nope. Sorry.
Looks even better.
It's decided to get finicky here. Yeah. Oh no. I'm sorry, guys. Let's see. There we go. Okay. So same thing that we did last time. We're going to start by refreshing our memory of inheritance and Mendelian genetics. So please, in your chat box, type as many things as you can remember relating to inheritance or Gregor Mendel. Okay. We're going to give you just a minute or so to do that.
I'm playing along too, Ms. Cox. Since I'm a chemistry teacher, I'm allowed.
Yeah, absolutely.
Wow, this is hard.
It is hard.
Ah, nice job, Libby. You got a lot more than I got.
Oh my goodness. Okay. Having a little Zoom difficulty this morning. All right, let's read through some of these. Go ahead and submit what you have if you've not already done so. Let's see. Independent assortment, dominant genes. Oh, good job, guys. I'm seeing a lot of the vocabulary that we hope to see when we talk about heredity and genetics. And this is one of the more vocabulary rich portions of the biology content, I think. And if you're wondering of a quick way to boost your score as we prepare for the free response questions, I would say study those vocabulary terms. And there's lots of Quizlet sets that are already ready for that. So I think that's an easy way to start getting ready. You guys are doing such a good job. Okay, so I see that we're mentioning a lot of good things. So here's the next thing I want you to do, and we're going to do this on a poll, so it's anonymous. I want you to rate where you think you are for Mendel and inheritance. So this would include everything from like chi-square to predicting the ratios of inheritance, pedigree charts, anything like that. So rate where you think you are on a scale of 1 to 4, 4 being very confident.
Hi, Trevor. Yeah, I know it goes 1 through 6, but just use 1 through 4. This is a general all-purpose polling. And we've got about half of you chiming in already. So everyone else, let us know where you stand on Mendelian genetics. And teachers can chime in too, and don't feel like you have to be at number 4. Yeah, you can answer as if you were a student. Okay, Brittany, I'm going to close the polling here. And it looks like most of us feel like they're in the 2 to 3 range, so a little bit of familiarity.
Yeah, I think that that's a pretty good place to be. I'm really excited that there are no 1s. So as long as we have no 1s, I think we're in good condition for the test. All right. So I want to introduce you to this guy. You've probably seen his face. This is Gregor Mendel, known as the father of genetics. And I find it very interesting to drop these scientists into history, because I think when we reflect on them independently from the other things taking place in the world during that time. So, okay, so he did the bulk of his work from 1856 to 1863, and he published in 65. Can anyone think of a major event that was occurring during that time period? Maybe some of my juniors who take U.S. history. Brownie points, right? He was working during the American Civil War. And I find that very interesting because we tend to, the Civil War. Good job, Trevor. We tend to think of Gregor Mendel as way back when, but he was actually more modern than we like to think of him. Good job, guys. Your brains are on this morning. So he gave us 2 main laws, and some of you mentioned those as we were going through the first question. He brought us the law of segregation, which means traits are particulate. They don't necessarily blend together. And the way I think about this is if you were mixing 2 different colors of M&Ms versus 2 different colors of paint. If you mix 2 different colors of paint, you get a new 3rd color. Whereas if you're mixing 2 different colors of M&Ms, they just make a mixture. So it's particulate like the M&Ms. And when you hear law of segregation, I want you to think of alleles. Like that's the association you should think of with law of segregation. And the other one he gave us was law of independent assortment. So allele pairs separate without condition from other pairs. We know now that not everything follows Mendel's rules, but these were great rules for when we started. So when I say Mendel, I'm sure many of you think of Punnett squares, right? And most of you have probably done the basic Punnett squares that are 4 little boxes with one trait. Did any of your teachers have you do the dihybrid crosses with 16 different squares where you look at 2 traits at a time? Just in the box, yes or no? Did anyone have the dihybrid crosses? Yes, yes, yes. Yeah. I can tell you from experience that the dihybrid crosses are not a major favorite among students. They're a little tedious. And if you're trying to look at 3 traits in combination, then the boxes get even bigger and it becomes a very stressful situation. So I'm going to talk to you guys about the law of multiplication, which is a faster way to find out the combination of traits given parental genotypes. And it's a lot easier than the dihybrid crosses. I promise. So this is just a basic probability rule. If you multiply the likelihood of 2 random events occurring, then it'll give you the likelihood of them occurring in combination. So for example, the likelihood of flipping 1 coin and landing on heads is 1/2, but the likelihood of doing it twice would be 1/2 times 1/2, which is 1/4. And even though not everyone loves fractions, we love them more than dihybrid crosses, right? So here's what I want you to set up and take just a second. If round seeds are dominant to wrinkled and tall plants are dominant to short plants, what is the likelihood that these 2 parents will have a short and a round offspring? So set that Punnett square up. See what you can come up with. It should go a lot faster with the law of multiplication.
Are you making a video? Are you on FaceTime with a bunch of kids? Can you see the kids?
And go ahead and put your answer in the chat box when you've got one.
So to do this, you would set up 1 Punnett square with the round versus wrinkled trait, and then 1 Punnett square with the tall versus short trait.
I see a consensus, but remember round is dominant to wrinkled.
Yep.
So I think if you have 1/8, you might be thinking wrinkled are going to be dominant to round.
Yeah.
If I know my Punnett squares, which I'm not sure I do.
Yeah, you're right. So as Lou said, if you're doing 1/8, go back and look at your round and wrinkled Punnett square. All right, Lou, you want to share the answer with them?
Well, it's all on the chat, Brittany, and we've got a lot of 1/8 and a few 1/4. Aaliyah Frisbee, good God job. I think 3/8 is the answer I got, and I'm not swearing that that's correct, but will you walk us through this one, please, Brittany?
Yes. So to set up the round versus wrinkled would be the first one, and you set it up like a basic, monohybrid cross. So I would say that 3/4 will be round because that is the dominant trait, because the heterozygous zygote as well as the dominant homozygous offspring will have that round trait. And then I see that 1/2 of the offspring are going to be short. So 1/2 times 3/4 is 3/8 by my math. Does that make sense? Now, the reason I really wanted us to do this together is not because I doubt your guys' ability to multiply fraction. Thank you, Austin. It's that I think that this shows one of the problems with the test is when we're reading these long strings of text, we don't always pick out the correct facts. And we make easy mistakes like the round versus wrinkled that Lou pointed out for you guys. So during the test day, what I would suggest is you have scrap paper available so that you can write down key facts like round equals D, like dominant or whatever, just to help you keep it straight and make sure that you're reading actively. Because when we get a little stressed out, a little nervous, it's really easy to make mistakes in reading it. All right. How many of you guys have seen this before? Anyone seen the chi-square statistic before? Yeah. Good. So chi-square has been a favorite when we had math questions. It could show up on the FRQs. And to find the first of all, what's the purpose of the chi-square value? Why are we looking at chi-square? We're looking at chi-square to determine if something's due to random chance or if there's something else going on. So if I flip a coin 10 times, how many heads would you expect if I flip the coins 10, if I flip a coin 10 times? How many times should I get heads? 5, right? So if I got 6 or if I got 4, that wouldn't be necessarily questionable. But if I got 10 heads, you might think that I have a weighted coin, right? Something's going on. So that's what chi-square in its simplest form is telling us is it within a reasonable range of what we expected, or do we think something else is in fact going on? Okay. So I'm not going to go through the example super specifically unless that's something we need to do. But what I want us to be clear on is that when it comes to genetics problems, the expected value is coming from your Punnett squares. Okay, so your expected value is coming from your Punnett squares. So for example, if we were doing a chi-square on the outcome we just had, I would take the number of offspring times 3/8 to find my expected number of offspring who would have been round and short, okay? So the expected numbers are coming from the Punnett squares. The observed is whatever the experimenter sees, okay? And then I have all the steps in here because I'm going to make these slides available to you guys so that you can go through and study it at a more leisurely rate. But it's really tedious where I can't show you guys, like write it out. I can just talk through the steps. It's more difficult to go through the math problems together, but I wanted to make sure that you had a good example available. Maybe the next time I do this, I can have a document camera set up in my house. I don't know.
And I don't think I would be shocked if the AP exam, particularly this year, asked students to perform these calculations, but I would not be surprised if they were expected to interpret them. So they might be given chi-square numbers and asked what they mean. And that's, I think, the important part of this exercise.
So in that respect, the chi-square number, what you do once you calculate it is you compare it to your critical value, okay? And that's what this table is showing you, is it the critical values based on the degrees of freedom. So if your chi-square value is less than the critical value, you accept the null hypothesis. So what in fact is the null hypothesis? What does it mean to be null? N-U-L-L, null hypothesis. Null hypothesis means that everything's normal. So that would be like the 4 or the 6 coins instead of the 5 heads. Yes, no significant statistical difference. And make sure that you include that statistical phrasing. So if your chi-square value is less than the critical value, then you accept the null hypothesis, meaning that there's nothing going on. Okay, if it is greater, yes, no statistical difference, no significant statistical difference. That significant word is important too, okay? So if you are greater than the critical value, you reject the null hypothesis, which means there is something else going on. So there's one major culprit in genetics that's generally behind, or is often behind, rejecting the null hypothesis. So can anyone think of something that happens in genetics that causes us to see distributions of traits that are not what we expected? Distributions of 2 traits together that are not necessarily what we expected? Yeah, you guys are so smart. You're doing such a good job. Gene linkage, and you can, I hope you think of little fruit flies when you hear that, and Thomas Hunt Morgan. So anytime you see that the actual numbers are not matching up with your accepted and you're rejecting that null hypothesis, I want you to think about gene linkage as a possible solution, because that's kind of the low-hanging fruit there. That's the classic example. I do have some resources linked here for you, and the AP College Board has already done their reteach on this topic, and their Mendelian reteach focused on the chi-square. Okay, so that was their focus, was doing chi-square calculations. So if you are not very comfortable with that, please go back and catch these resources. And you can always contact your teacher, or you can contact me and we can work something out. All right, you guys ready? All right. First question, we're going to give you a poll in just a second and see what you guys come up with.
Yeah, they're pretty quick on the calculators.
So there is a tool that I hope you went to. What tool would help you answer this that you should have out during the exam? Your formula sheet, right? It has all the critical values on it. Look over that formula sheet. It has lots of good information that can help you find your way out of sticky questions. All right, we've only had about 9 students vote, so I'd like to see a few more come in before we move on to the solution. We currently do not have a favorite among any of the answer choices.
Now let's give a couple
I'm
of students
sorry,
a few more
Julie.
minutes. And Brittany, you may have to go through this one. All right, so and I think a few of the screens don't see the poll, and I don't know why. I'm clearly not a computer scientist, so this may be all we're going to get. So I'm going to end the polling in 3, 2, 1 right now, and we get about half the responses. So Brittany, I think you might need to go through this one for us, for me.
You're split between A and C. So A is the correct answer. Sorry. The poll is still popped up in my way. So this is the critical value with 1 degree of freedom. Okay. So the trick here is there was no calculation. There was no calculation. You were just looking at the formula sheet. Okay. You knew there were only 2 alleles for the trait, which is 1 degree of freedom. So all you had to do was refer to that critical value table in order to answer this. And that's really stressful because we overthink things, right? So this should have been a very simple question, but when we saw it, we automatically thought, I need to calculate something, but there's not enough information really to get there. So all you needed to do was say there's 2 alleles, so that's 1 degree of freedom, and look at your critical value table. Does that make sense? Tricky, huh?
Very.
They are a little tricky. The AP readers or the AP writers, and no offense to Lou, but I always tell my kids that they're kind of cruel and unusual and they look for new ways to trip us up. Yeah, Trevor, I think that that's pretty common, is that chi-square is a little rough. And I did link several resources so that you could practice up on that. All right, let's look at this question, and Lou's going to give us another poll.
I'm going to give folks a chance to read this question first.
I'm sorry if you guys can hear my kids in the background. On day whatever of NTI, they're a little restless.
Okay, I've finished reading, so I'm going to launch a poll. And the polling is anonymous, students, so don't feel like you're going to be chastised. I think it's best to take a stand here.
They're tough questions.
Unfortunately, it won't let me vote, Brittany, so I'm going to go out on a limb after I end the polling in 5, 4, 3, 2, 1, and there we go. I'm going to end the polling now and say that I would have polled for answer D, and I'm not sure that's right, that was a popular.
D, correct. So answer choice D is correct, and that was very reassuring. Lots of you guys picked answer choice D, okay? And that is the classic ratio that you guys have probably learned with dihybrids, but you could also throw your law of multiplication in here if need be. So the other thing I want to share with you guys is the questions. Yeah, I think it was a little easier, Trevor. The questions that we're going to go through today, they are all released AP questions. So I think sometimes things like law of segregation seem simple on the face of it, and we think we understand it really well, but then when we get into the actual AP style questions, they ask the questions in a way we're not prepared for. Okay, so that's why I really wanted to focus on going through some of these questions today so you could kind of get a feel for how the questions may be phrased, okay? All right, other types of inheritance. I gave us some pictures here. Incomplete dominance is when the two alleles both affect the phenotype. So you get an intermediate or a middle phenotype. And this is like the paint mixing example that I shared with you guys earlier. The second one is co-dominance. Any of my ag kids know what kind of cow this is? What would you call this coloration, I guess, on the cow? Yeah, I taught in Lincoln County. I get the ag kids a lot. So this is a roan coloration pattern, yeah. And it's roan because the cow has some white hair and some red hair. So this is co-dominance. The hair is white and red. It does not blend to a pink, although I think a pink cow would be pretty okay. Like I would be for it. And then the last one is sex-linked inheritance. Classic examples of sex-linked inheritance, hemophilia, colorblindness, right? Usually we talk about X-linked inheritance, but sometimes there is Y-linked inheritance as well. But the Y-linked inheritance is less likely because there are fewer genes on that Y chromosome because of its diminished size. All right, here's everybody's favorite, the pedigree charts, okay? So if you've seen a pedigree chart, circles are women, squares are men. The generations are numbered with Roman numerals, and then the individuals are numbered with 1 or 2. So for example, this male would be I-2 because you number left to right. So I think that's tricky because they refer to them in those numbers, but if you've not practiced naming them that way, you can't really navigate the diagram. So just quickly in the chat box, what would you name this female? What would this female's name be on, well, you can't see my cursor, can you? Can you see? Oh, you can? Okay. What would this female's, yeah, this one right here. But if you named her for the specific female, so she is a carrier, good job, but what would you name her using the Roman numeral and then the number? So she would be individual. Roman numeral 3-2, okay? So she would be individual Roman numeral 3-2. So I'm going to give you one more to see if we can read this pedigree chart. And I want you to look at individual I, Roman numeral 2, right? -2, okay? And tell me if that individual is affected or unaffected. So I want you to look at individual Roman numeral 2-2. Is that individual affected? Yeah, good job. Good job. Yeah, they're affected, right? They're filled in, which means that that person does have the phenotype. And you guys also hit it very well that this partial shading indicates a carrier. You also, it's not always a dot in the middle, that's kind of odd, but sometimes it's a half shaded circle, which also indicates a carrier or a heterozygote. All right, so I want us to look at this pedigree chart together. And I want you to look at this. Do you think that this is autosomal dominant, autosomal recessive, or sex-linked dominant or recessive? This one's really tricky. Okay. So lots of you guys are saying it would be sex-linked recessive, but if that were the case, affected mothers would have to have affected sons, okay? Because mothers to be sex-linked recessive would have to have 2 of the recessive copies of that gene and must pass 1 to the son on the X chromosome. And because of this individual, we know that it cannot be sex-linked recessive, okay? So I would say we know it's definitely autosomal. Okay, we know it's definitely autosomal. So do you think it's autosomal dominant or autosomal recessive? It's dominant, right? If it was recessive, then we would not be able to have this one over here. So it's a dominant trait. Good job. You guys did a really good job with that. Pedigrees are tough. All right. I don't know if you guys have seen these guys before. They always make me think of Lady and the Tramp, but this is a Siamese cat. And Siamese cats are a great example of how the environment and your genes interact to cause the phenotype, okay? So the Siamese cat, it has this really unique coloration where the muzzle and the ears and the paws are dark, while the lighter part of the cat's body is light. And this is due to something called point restriction. So there is an enzyme that determines coloration, and it's only active below certain temperatures. So the core of the cat is warmer than extremities, right? The ears are going to be cooler. So the pigment is able to be deposited in the ears, muzzle, and paws, whereas the pigment is not able to be deposited on the cat's body. So that is a good example of gene and environment interaction. Can anyone think of another one? There's one more big classic, and that one, this one's point restriction. The other classic is hydrangeas, right? Because the pH determines the pH and the genes determine the color. All right, epistasis. Epistasis is another one of those vocabulary words that we have to consider when we're looking at genetics. Epistasis simply means, it's a difficult word, that means more than one thing can control the trait. Okay, so more than one gene is controlling the phenotype, and that is epistasis. All right. One of the classic examples you can think of is albinism, because especially with the eye color, that person would still have a gene for eye color from their parents. Like they would either have normally blue or brown eyes, but there is another gene that's interfering that inhibits the depositing of that pigment. So that's epistasis, that the one gene allows no pigment to be deposited, so it doesn't matter what color pigment they're coded for. Does that make sense? A little bit? Okay. All right. Hybrid vigor is, if you think about hybrid vigor, the explanation is actually really complex. That's why I put this actual excerpt from the textbook on here. For our abilities today, for our purposes today, we're just going to say that hybrid vigor means the heterozygote is more successful than the homozygotes. Hybrid vigor is especially used in agriculture. They use it a lot about grain, corn, like which wheat species produces the most grain. I'm not an ag person, but that's where this really comes out. A lot of flowers. So anytime you see hybrid, you think heterozygote, and vigor means that it is succeeding, like it's very vigorous. So hybrid vigor is when the heterozygote is more successful than the two independent phenotypes. All right, here's my resources. I hope you guys find that helpful. I know that we can only go surface because an hour is not very, very long. All right, you guys ready? Okay, Here we go.
And I'll go ahead and open up a poll here.
Thank you.
And move that out of the way.
Lou, can you guys just see the PowerPoint, or is the chat box like in the way?
You can move the chat box, I think. I mean, I can move my
I mean,
chat box.
is my chat box in the way? Can
No,
you guys
we cannot see your
okay.
chat box.
Okay. I didn't think so, but I just wanted to make sure. All right, guys, let's lock in an answer. Go with your gut.
Okay, we've got just about half the participants responding here, and we're getting a few more votes. I'll give you about 5 more seconds.
Try to get it down to 50-50 and then just commit one way or the other. Yeah. Like eliminate some answer choices.
These are anonymous, so don't be afraid.
Yeah, don't be afraid.
Okay, and there we go. So we had a late rally for choice D, mitochondrial gene.
Yes. Survey says.
Which was not what I was going to guess, so I'm not going to say that. Oh.
Right. So the reason we can tell is all mothers' children are affected. It doesn't seem to be dependent on father phenotype as well. So if the mother has it, the kids have it. If the mother doesn't have it, the kids don't have it. And that's because we get our mitochondrial DNA from our mothers due to the extracellular inheritance that happens during fertilization. You guys are good. I'm trying to trip you up.
You got me on
Here
that
we
one,
go.
Brittany.
Wasn't me. It was the AP writers. I'm sorry.
I'll put up a poll for us now. If we've
You guys have some serious confidence on this one, and I like it.
I think we're getting better. The responses are coming in much quicker.
Yeah.
Or maybe this was an easier question. I want to see figure 2.
Yeah, we will.
Oh, good.
I didn't have room for all of them on the one slide, but the one that
That's
needs
okay.
figure 2, we have.
I'm fascinated by the relationship between, Mendel and the laws of inheritance and what we now know about DNA and DNA replication and operons controlling genes and things like that.
It's really beautiful when you see the whole picture.
Okay, we've got about almost 3/4 of our participants. So I'll end the polling in 3, 2, 1. There we go. And it looks like the popular answer was A, autosomal recessive, which is what I was gonna.
Correct, right? Yeah. If you have 2 parents who are unaffected with children who are affected, it has to be recessive. All right. Let's look at this one. So it's the same stimulus.
Oh, I've got an answer for this one already.
We have quite a few votes already. That's good. You guys are
We
good
do,
at that.
and I'll close the voting in about 5 more seconds. So if you're just about to choose, take a stab. 3, 2, 1, and looks like B is the popular choice here, which is what I would have had.
B is correct. Okay. Oh, sorry. I was trying to close my poll. Both parents have to be carriers. All right. So here is figure 2. And this is kind of biotechnology meets Mendel, but I wanted to put it in here just so you could see the connections, because that's really what the test is all about, is making connections from those individual pieces of content.
Now, do we need to refer to the now I forgot, I'm spacing on the name, the family tree for this?
You should just need figure 2.
Okay. Darn.
I think. But I'll go back and show you the other one as well. You may want to screen grab this pedigree. And then there's figure 2. Guys, 6 is not an answer choice.
I think you can change your vote.
Is that taking a knee? Like, I don't know. 6.
Hmm.
As you come across, well, you guys aren't doing multiple choice, so you don't need my multiple choice strategies. But I have them.
This one's a little bit trickier, so I'm going to keep the poll open for about 10 more seconds. I've got a choice here. Okay, 3, 2, 1, and there we go. So it looks like C was the that was not a popular answer. And I was torn between A and B. So Brittany, perhaps you can walk us through this one?
Yes. Oops. Come back. So the big piece here is reading this gel electrophoresis. So we know that individual 3-4 has the same alleles. It's a homozygote, right? Because it has one thick band at 500. So those alleles both have a weight of 500, those fragments. So we know this one is a homozygote. We also know that 4-1 is a homozygote. Sorry, guys. A phone call came in and it's ruined everything. Can you see it again now?
We can still see.
Okay, good. And so if there are 2 discrete bands, that means that there are 2 different alleles and you're seeing them show up here. So both individual 2-2 and 2-3 carry 2 different alleles.
And Brittany, I guess
We'll
my
get you down.
question, because on choices A and B differ only in identifying individual 4-1 as having 2 copies of the mutant allele or the wild type allele. And I missed how you would know which of those alleles has a larger DNA fragments. Was that somewhere in the question? Oh, caused by an insertion mutation.
It was not,
I see that now.
Yeah.
I completely skimmed over the word insertion, which seems pretty critical in deciding that choice A is better than choice B.
Yeah.
Wow,
Right,
that's a lot.
because
There's
the
a lot of biology in
insertion-
that question.
There is, and the questions are super elegant in the fact that they can find a way to test so much content with one little string of text.
I guess you have to be careful to read the questions carefully. Now, of course, there will not be multiple choice questions on the exam that you students will be taking on May 18th, I believe, but the understanding is going to be there, and you'll still need to read carefully.
Well, I think in previewing mock exam responses, this is a trend we even see in the FRQ writing is you guys start to read the stimulus and then you kind of see what you want to see. So make sure you're reading exactly what it says and not what you anticipate so that you can answer the question that the writers are going for. And then here are some more resources. I think that this, especially on chromosomal inheritance and environmental interaction, may be helpful because it'll go back through meiosis and Mendel, which is valuable. But if we had done meiosis and Mendel, that would have taken most of our time. And that's just not, choices weren't made. It was a sacrifice.
No, and I think that our hour is almost up, Brittany. So I think this is actually a pretty good place to stop, if that's okay. But we will be back.
Okay.
And I think this is a good idea to remember that the AP exam this year will be open note, in fact, open book. So we'll be back after to look at the mock exams and maybe have students thinking about what they should have around them when they're actually taking the AP exam. It will not be a lot of factual knowledge, stuff that you can look up in a book, because we don't care about whether you can do that. It will be applying these principles and being able to phrase and good explanations for them.
I think critical reading, like we've been talking about, how the question can be tricky, is going to be paramount this year as well, because for them to get to that real tough application level, then there tends to be a lot of intricacy in the text. So I think that really paying attention to what the question says is going to be so important for us.
Well, good reading skills are important no matter what you're going to do. So before we sign off, let me remind particularly students from Barberville High School that make sure you have logged in and put your name and school in the chat box so that Ms. Hubbs will know that you were participating, and welcome to all of you. And to all of you other AP Bio teachers and students. And we'll hope to see you one more time as the students, when we review the mock exams, and Brittany will show some sample responses and let you have a crack at scoring them. So have a good afternoon, and I will see many of you later virtually, if not in person, soon enough, I hope. Thank you so much.
Thank you, guys.
I'll keep the chat box open for one more minute, so please do go ahead and make sure your names are in there.
You guys did great. I'm so confident in you. Just go back and brush up on what you're